So our next topic is carbohydrate molecules that are conjugated to other types of macronutrients, in this case, proteins. So this is going to be a lot of definitions and description. And our topics are going to include glucosamidoglycans, or GAGs, proteoglycans, glycoproteins, and lysosomal storage diseases. Our learning objectives will be to define glucosamine, indicate food sources and clinical implications, discuss, again, glucuronate, oh wait, that's a typo, glucuronate metabolism and its clinical significance, list and discuss glucosaminoglycans, indicate locations and their clinical importance, list and discuss some of the MPS diseases, and define glycoprotein and indicate their clinical significance. Okay, so first I'm just going to start out with some simple definitions. Okay, so glucose can be combined with other molecules, carbohydrate or not, to form more complex molecules with specialized functions and properties. And some of these complex molecules can have very specialized functions. So a really simple example of this is an amino sugar, such as glucosamine. And this occurs when the C2 of the hydroxyl group on a glucose is replaced by an amino group. Then we have glycoproteins, in which proteins are covalently bonded to a carbohydrate. We have glucosaminoglycans, which are an unbranched polysaccharide consisting of alternating 2 units of different sugars and are often located in the extracellular matrix. And then we have proteoglycans, which are large molecules made up of proteins and glucosaminoglycans. Okay, so amino sugars are important constituents of glycoproteins and glycosaminoglycans. So we're going to go through each one of these in this lecture. So the first molecule I want to talk about is glucosamine. And this is a simple amino sugar that's manufactured in the body from glucose and amine. It's a major component of chitin that we find in the exoskeletons of animals such as crabs and lobsters, and one of the primary physiological roles in joints is that it stimulates the manufacture of glucosaminoglycans, which are a key structural component of cartilage. So glucosamine promotes the incorporation of sulfur into the cartilage, and therefore when glucosamine is paired with sulfate, it's thought to be a better source or a more effective source of glucosamine for treating osteoarthritis clinically. Okay, so we produce glucosamine in our bodies. There's really no food sources. As I said, it is in the exoskeleton of some animals, but we don't typically eat those for food unless they're isolated and put into a supplement commercially. So the deficiency of glucosamine, when our body does not produce enough of this, or we have excess deterioration of the cartilage, we see that deficiency is a major factor in developing osteoarthritis. So the joints that are most affected by glucosamine deficiency are the knee, hips, and hands. And deficiency causes cartilage destruction, hardening and formation of large bone spurs in joint margins, and pain, deformity, and limited joint motion. And morning joint stiffness is often the first symptom. So we can see in this graphic here, we have a healthy knee joint. Our cartilage is intact and working and providing support for the bones at the joint. And here, though, we see that we have the deterioration of the cartilage. Therefore, we don't have this kind of cushion between the bones, which causes spurring of the bone, and then we have erosion of the cartilage. So glucosamine is thought to be a safe and alternative, effective to aspirins and other non-steroidal anti-inflammatory drugs. So NSAIDs are fairly effective in suppressing symptoms, such as pain and inflammation, but they can possibly worsen the condition of osteoarthritis because they themselves can inhibit cartilage formation or accelerate cartilage destruction. So if a patient who has osteoarthritis is taking NSAIDs, there might be short-term relief from the decrease in the symptoms, but long-term exacerbation because it's inhibiting cartilage. They're inhibiting cartilage repair and inhibiting glucosamine-glycan synthesis. And we'll talk about more about the glucosamine-glycans coming up. So glucosamine sulfate relieves the problem at the source and helps the body to repair the damaged joints while also decreasing the pain and inflammation. So the standard dosage is 500 milligrams 3 times a day, though obese patients do need a higher dosage, and it should be calculated based on their body weight. And also, patients taking diuretics may have higher losses and therefore may need increased supplementation. So this glucosamine does have a good safety record, though there are kind of the standard side effects that we get with many drugs, including GI or stomach upset, heartburn, diarrhea, nausea, indigestion, kind of all of the typical problems. Okay, so our next topic is going to be glucuronate. And this molecule is derived from glucose in the uronic acid pathway. It's formed by the oxidation of the alcohol on the glucose carbon 6, and it's oxidized to an acid by an NAD dependent dehydrogenase. So again, it's taking the hydrogens off and therefore oxidizing it. So it's formed from the degradation of inositol, which is a second messenger for many hormones. And it's a highly polar molecule that is a constituent of proteoglycans. So this molecule combines with bilirubin and steroid hormones and helps with the excretion of bilirubin. And it can also be combined with certain drugs to increase their solubility. Okay, so here we have the pathway for the synthesis of glucuronate. So we're starting with our G6P, which, as we've talked about in our last lecture, these first reactions should be very familiar. Transformed to G1P through phosphoglucomutase, and then in an energy requiring step is then converted to UDP glucose, which then, with the dehydrogenase that I just mentioned in the last slide, is converted to UDP glucuronate. And this is a precursor to glucuronides, proteoglycans, and glucosaminoglycans. So glucuronate can also be transformed to L-ascorbate or vitamin C, but not in primates. Okay, so in the adult human, a significant number of erythrocytes die each day. And erythrocyte degradation releases significant amounts of the iron-free portion of the heme porphyrin ring, which is degraded, yielding bilirubin. Okay, so in the bilirubin, in the liver, bilirubin is solubilized by conjugating it with glucuronate to bilirubin diglucuronide, which is then secreted in the bile. Okay, so let's go back to our last slide here. Oh, sorry. Let's go to our next slide, actually. Okay. Okay, so here we just kind of have the normal process for excreting the bilirubin, which is the degradation product of the erythrocytes, okay? So we have our blood, it's degraded, we have to bilirubin. Bilirubin then goes into the liver hepatocytes where it's conjugated with glucuronate, and then it goes through the biliary system into the small intestine and then is excreted into the feces, okay? So if we don't have the ability to conjugate bilirubin with this glucuronate, this contributes to the buildup of bilirubin in our system, which contributes to jaundice. And we can see this in often in hepatic disease. So the level of bilirubin production exceeds liver capacity. Okay, so next let's talk about glucosaminoglycans, or GAGs. And these are the most abundant heteropolysaccharide in the body. Okay, so they are, okay, so they're long, unbranched polysaccharides with repeating disaccharide units. So they contain a sugar, and one of these two that's listed here, and a uronic acid, either glucuronate or another derivative, iduronate. And here's a simple glucosaminoglycan here with glucuronic acid and acetylglucosamine repeating. These GAGs have a highly negative charge and are located primarily on the surface of cells or in the extracellular matrix. Oh, and just as a reminder, the uronic acid is a weak sugar acid, which is a monosaccharide derivative. So instead of its CH2OH, it has a COOH. Okay, so glucosaminoglycans are very viscous, and so they have very low compressibility. So they're so thick that you can't really compress them, and therefore they're ideal for lubricating fluid in the joints because they don't allow the bones to compress right against each other. Also, though, they have some rigidity, which provides structural integrity to the cells and provides a passageway between the cells, which is important for cell migration. So there's a number of GAGs with physiological significance listed here: hyaluronic acid, dermatan sulfate, heparin sulfate, and keratan sulfate. And the location of these GAGs are listed here. We can see, though, a lot of them are in the extracellular matrix, loose connective tissue, cartilage, valves, bone. So you can see these are really important structural molecules throughout our body. Okay, so briefly, I just want to touch on hyaluronic acid. And this is a unique GAG in that it does not contain sulfate, and it's not found covalently attached to proteins in a proteoglycan. And most GAGs are, the main purpose of a GAG is as a constituent in a proteoglycan, which we're going to, I think, talk about next. But for this GAG, it's not found attached to proteins in a proteoglycan. And these polymers are very, very large and can displace a large amount of volume, and therefore are excellent lubricators and shock absorbers. So we can see these are in our synovial fluid and in the extracellular matrix of our connective tissue. Okay, so next we're going to talk about proteoglycans. And these are large, complex macromolecules that consist of a protein core to which a GAG is covalently linked. And these are also called mucopolysaccharides, or MPS. So these are synthesized in the lumen of the endoplasmic reticulum and in the Golgi apparatus. And in a proteoglycan, we have a core protein, and then we have GAGs that are extending perpendicularly from the core as if they were a brush. And the linkage between the GAG and the protein involves a trisaccharide with 2 galactose residues and a xylulose residue. Okay, so why do we care about GAGs and proteoglycans? Well, these molecules perform numerous functions within the body. So, for instance, the GAG heparin prevents blood coagulation. We've heard of heparin a lot. It's often administered in certain surgical procedures in order to keep the wound open and prevent clotting so that the physician can continue to work on the patient. So in the body, it's naturally found abundantly in the granules of mast cells that line blood vessels. And injury initiates the response to release the heparin from the granules, um, which leads to inhibition of blood clots. Now, there are several genetically inherited diseases that result from defects in the lysosomal enzymes that are responsible for the metabolism of GAGs. And these are termed mucopolysaccharidoses. So we'll remember that that's in reference to the mucopolysaccharide, which is another name for a GAG. And because the patients with the lack of these enzymes are unable to break down the GAGs, it leads to an accumulation of GAGs within the cell. And we see this in lysosomal storage diseases. So there's a lot of different types of diseases that can affect GAG metabolism, and here's some of the most that are well known: Hurler syndrome, Hunter syndrome, etc. And all of these disorders, except for Hunter's, are inherited in an autosomal recessive. Okay, so we'll just kind of briefly go over these. So we have type 1, which is Hurler's, and here the symptoms are listed here, but we can see some of the things that are kind of common to the last lecture we talked about when we have this accumulation of glycogen. So we have the organomegaly, because we're not able to break down the GAGs, dwarfism, because we're not able to utilize nutrients, mental retardation, early mortality. For type 2, that is Hunter's type. It can be mild or severe, but again, we have this organomegaly, but we also have facial and physical deformities and mental retardation and early mortality. Type 3 is inability to break down heparin sulfate and results in profound mental deterioration, again, and the various tissues that are impacted. And, oh, I'm sorry, I said, yes, that was right. Okay. And then again, we have type 4, which leads to, again, this hypoplasia, aortic valve disease, and skeletal abnormalities. So there are more types than this, but these are kind of the most common or well-known types. Okay. So next, let's move to glycoproteins. Okay, so the distinction between, it gets kind of confusing when we're talking about proteoglycans or glycoproteins, but the distinction between these two is basically the level and the types of carbohydrates that are modified. So with the proteoglycan, we had kind of this protein core and the GAGs coming off of it. And in glycoproteins, we just have a protein that is covalently linked to a short carbohydrate chain. Okay, and they are linked to either the serine, threonine, or asparagine residues in the protein. So these are most of the proteins that are in the blood have been glycosylated into glycoproteins. And we see a lot of glycoproteins on cell surfaces, and we can see that in this graphic here. So this is the lipid bilayer of the cell membrane, okay? Here we have the protein that we're talking about that was referenced here, okay, and that is both on the cell surface and the cell intercell. I guess I'm sorry, I'm trying to get my arrow here. And okay, goes through the lipid bilayer with its hydrophobic portion, and then also has an intercellular portion. And so you can see on the outside of the cell, we have this carbohydrate moiety that's attached onto the protein. So when glycoproteins are on cell surfaces, they're really important for a number of reasons. Not only to maintain the structure of the cell membrane, but also for communication between cells. So this carbohydrate moiety is often used as a type of recognition signal for many other types of molecules. Okay, so an example of this, of a glycoprotein, is the A, B, and O antigens in our blood, which are carb moieties of both glycolipids as well as glycoproteins. So several viruses, bacteria, and parasites have exploited the presence of these cell surface carbohydrates, usually glycoproteins, and use them as portals of entry into the cell. So we can see this in this graphic here, where we have a protein, we have the carbohydrate moiety, and then we have a number of harmful, usually, type of cells or other harmful molecules that attach onto these carbohydrate moieties and use them to access the cell. So one example of this is HIV, which gains entry into the cell by interaction between the surface glycoprotein and the CD4 cell host receptors. Okay, so this was kind of a short lecture. Here are your practice 2 questions, and of course, feel free to email me with any questions. Have a good day. Bye.